right, so what we're talking about, so we just went over a little bit about Mendel. What we're going to be talking about is the idea of the principle of segregation, or what we call a monohybrid cross or a single trait cross. Now, looking down here, this is figures in your book, of course. Let me see if I can get it in focus here. Looking here, it's not really in focus, is it? It's okay, I guess. All right, so you can see that there's traits that Mendel looked at. These were the garden pea traits. He was looking, what we call the dominant trait is the purple flowers, for example, and the recessive trait there is the white flowers. And you can see through this table there's a bunch of traits. So you should probably look these over and know kind of generally what they are. Round-seeded is all the dominant ones. Just remember one side, and that helps you, you'll know the other side is not dominant. Round-seeded, yellow-peed, axial flowers, inflated pods, green pods, and tall plants. So purple, tall, round, yellow, those are really common ones that people use to talk about Mendelian traits. All right, so let's look at this. So what he did was he took these flowers, these true breeding flowers, let's say of a purple plant. This is what he called the parent generation, or what we call the parent generation now anyway. And he took purple flowered true breeding plants and crossed them with white flowered plants. And like I said, he was doing the crosses manually. And then in the first generation, or what we call the F1 generation, all of them came out to be purple. So he assigned two terms to those, dominant and recessive. That is, in this first generation, they were all purple, so that meant what happened to the white, or the white went under and the purple showed. So if the white was still there, then that meant that purple was dominating over the white. At this point, he wasn't really sure that the white was still there. So then what he did was, his question was, is it still there? So he took the F1 purple flowered plants and crossed them with other F1 purple flowered plants. So this was, the next generation of peas. And what he got was about 75% purple and 25% white in the second generation, or what we know as the F2. So his question was, how could that possibly happen? So he knows it's not just one factor. In other words, you can't just have one purple factor and one white factor to get all purple. You could in the first generation, because what you have going there is that there's a combo. But then in the second generation, what's going on? If they're all purple, you could have purple, and they should be all purple, right? But instead, what he thought was, well, then there could be two factors. So he just assigned two factors to each one. So in this case, this is representing the purple flowers, or the true breeding, what they call dominant. We always write, just as a convention, capital letters, and I always use letters that you can tell apart. So I wouldn't be using P in this case, although that might make some sense because we're talking about purple. But little p is difficult to tell from big P, so I use A instead. All right, so anyway, this is the purple, two factors, making what we call a homozygous purple, or true breeding purple, crossed with a true breeding white. So this is a homozygous white. And in the first generation, so if I separate, like we do in meiosis, the two homologous pairs, because that's what would be happening, these would be alleles on separate chromosomes, or separate homologs, and they would separate during the process of meiosis. And in this case, no matter which cell I use for the separation, though, I'm still going to have capital A. And capital A is a factor coming from this individual. Likewise, over here, same thing. I'm still going to have, both alleles separating, but in this case, I only have one possibility, that is little a, and that is going to be coming here. So in the first generation, I'm going to have capital A, little a, or what we call heterozygous purple. All right, so in this case, This is really showing you the genotype of a heterozygous purple where the recessive trait goes under. All right, then in the second generation, what he did was he crossed F1 purples, which is that, times F1 purples, which is that. And in the F2, he got about 75%, 25%. So let's look at how. So in this case, again, the homologous pairs are going to separate. So in one cell, I'm going to have capital A, and the other cell, I'm going to have little a. And I can line them up as possibilities over here. Likewise, the same right here. And then when I cross them, I'm going to get capital A, capital A, capital A, little a, capital A, little a, or little a in my cross. And the results will be, I'll get 1/4 capital A, 2/4 little a, and 1/4 little a. This is what we call the genotype, or the genotypic ratio would be 3/4 to 1/4, 1/4 capital A, or that is 25%, or 2/4 capital A, that is 50%, or 1/4 capital A, or 25% for that. These are the genotypic ratios. All right. Now the phenotype is, so the genotype is what the factors actually are, and the that would be, of course, alleles, really. And the phenotype is like the outward expression. So in this case, purple and white. So purple, we would have as a phenotypic ratio, we would have 1/4 plus 2/4, that is 3/4 or 75%, versus 1/4 white, and that would be 25%. So that goes back to what he got in the F2 generation initially. So this was called the principle of segregation. That is, we call it the single trait cross now because we're just considering one trait. But what he was saying was there were factors that segregated from the individuals that were mating. And we know those are called alleles now. So these alleles segregate during the reproduction process, which is basically meiosis, right? Creating cells that are haploid, that are going to then recombine in the next generation or in the zygote, creating a combination or a recombination of the genes from each parent. So here we are